We are really fortunate to have Dr. Chantel Martin here to give us a presentation on her work trying to understand the molecular mechanisms underlying the social determinants of health in humans. Her talk is entitled Embodying Place, Social Environment, and Epigenetic Mechanisms. I've had the great pleasure of knowing Dr. Martin for the past few years since we overlapped at a conference, the IAPHS conference where we were presenting together. She got her undergraduate degree at the University of Delaware, then a master's in public health at UNC Charlotte. After that, she went on to do her Ph.D. at the University of North Carolina, Chapel Hill, where she stuck around as a postdoc, was awarded a prestigious K99R00 grant from the National Institutes of Health, and turned that into an assistant professor position at, you guessed it, the University of North Carolina, Chapel Hill, where she is right now. She just started back in June. Chantel's got some really great expertise trying to combine studies of human cohorts or human longitudinal studies, including the long-term adolescent health study and the Detroit neighborhood health study, which I think she'll tell us a little bit about today, and using those studies to really try and understand the molecular mechanisms, particularly the epigenetic changes that occur that are affected by these individuals' experiences. And I don't want to talk too much more about any of that because I know you'll get into it, but everyone please join me in welcoming Dr. Martin.
Well, thank you, Noah, for that introduction. Also, thank you for extending the invitation to present here at your seminar series. And also thank you all for coming. I'm always appreciative to speak and present my work to new audiences and receive feedback and insights from researchers from diverse backgrounds and interests. As Noah mentioned, I am an assistant professor at UNC Chapel Hill, and my work centers around identifying first the drivers and origins of health disparities, as well as trying to uncover the underlying biological mechanisms that link social determinants to health and health disparities, with a specific focus on epigenetic mechanisms. Like Noah said, I received a K99R00, and this is where I began to get some of the training and background and incorporating epigenetics into my work. The work that I'm presenting today focuses on the latter of uncovering the underlying biological mechanisms. And I realize everyone attending this presentation comes from different areas of research and has different baselines of knowledge. So I am going to spend a little bit of time on background and the motivation for the work that I conduct, and then get into some of the studies that my research group is currently doing. Okay, so first for the motivation behind this work. So despite improvements in medicine and technology, racial health disparities continue to persist across the life course, where Black Americans have worse health outcomes at almost every stage of life every stage across the life course than white Americans. And so here I'm showing in a table just some of these alarming health disparities across the life course. So starting here at birth, we see that Black Americans have higher rates of preterm birth and low birth weight, with low birth weight in the order of about over 2 times as likely to deliver a low birth weight baby than white Americans. Preterm birth, low birth weight, also risk factors for health later in life. So understandably and expectantly, we see that there are disparities in obesity in childhood as well as in adulthood, which leads to disparities in diabetes, hypertension, and even heart disease death rates. And what I want to point out here is that this is just, a handful of health outcomes that I'm showing here with disparities. But sadly, you can pull up almost any health outcome and see some disparities where Black Americans fare worse than white Americans. However, because of improvements in medicine and technology, we have seen increases in life expectancy at birth, particularly among Black Americans. Black Americans, we have seen steeper increases in the life expectancy. However, this is not done enough to really close the gaps or the Black-white gap in life expectancy. And we see that this is much wider still for Black males. So Black males have still lower life expectancy even today than any other racial gender group. And so behavioral factors, genetic factors have given us a little bit of information into some of the drivers of these gaps and disparities, but we have to kind of go a little bit further because we're still seeing wide gaps. And that's why we need to turn to some of these social and economic drivers of health disparities. And so particularly in the US, structural racism, and when thinking about the origins and where health inequalities begin, structural racism is a primary driver of health disparities. Zinzi Bailey et al. In 2017 provided us a working definition of structural racism in their 2017 Lancet paper, where they defined structural racism as the interconnected institutions whose linkages are historically rooted and culturally reinforced. It refers to the totality of ways in which societies foster racial discrimination through mutually reinforcing inequitable systems. This includes housing, quality, educational and employment opportunities, credit, healthcare, criminal justice, and so on, that in turn reinforce discriminatory beliefs, values, and distribution of resources, which together affect the risk of health outcomes. And so racial residential segregation is an example of structural racism that has had long-lasting implications. In the seminal work, public health paper that was published by Williams and Collins, they actually named residential segregation as a fundamental cause of racial disparities in health, and said that in order to have effective efforts to eliminate racial disparities, we must seriously confront segregation and its pervasive consequences. And so redlining is one of the most notable forms of residential segregation that was made possible by the Federal Housing Administration based on maps that were developed by the Homeowners Loan Corporation. This was over 80 years ago. And so these maps were created with neighborhoods outlined in red, in which they deemed them hazardous and risky for mortgage lenders and federal support, primarily due to the presence of minority populations, or specifically Black individuals and immigrant populations. And so this practice ultimately shaped neighborhoods as we see them today. Cities and towns all across the country use these color-coded maps to guide lending practices and maintain segregated neighborhoods. And so for context, I pulled some of these maps from somewhere near you guys, Phoenix, and I'm displaying the redlining maps from, over 50 years ago for Phoenix, where we see at Southern Phoenix, the areas or neighborhoods that were outlined in red and deemed as too risky for mortgages, too risky for lenders due to the presence of Black and immigrant populations. And actually, they also said they were either had high proportion of these individuals or at risk of having these individuals. And so now if we fast forward and look at the figure on the right, this is from the Robert Wood Johnson collaboration with VCU. This is in 2015, and we can see how the same area that was redlined and deemed hazardous, was not giving the resources to thrive, has lower life expectancy at 71 years still today. And this is in the order of 14 years shorter life expectancy than that of the northern and eastern area of Phoenix. And so neighborhoods don't just make up where people live, they actually shape every aspect of an individual's life. That's the educational opportunities, employment opportunities, access to healthy foods, parks and green space, access to quality healthcare, policing and crime, and even environmental exposures. And this is really what Zinzi Bailey et al. In their 2017 Lancet paper was describing as these mutually reinforcing inequitable systems. And these systems all work together to impact the risk of health outcomes and drive health inequalities. And so this has important implications for racial ethnic disparities, as people of color are disproportionately represented in these deprived, under-resourced neighborhood environments due to the historical policies like redlining that I previously mentioned. As a result, the consequences of this is poor educational opportunities, higher crime and policing in these neighborhoods, and limited access to nutrition, green space, and healthcare. Neighborhood environments and exposure can get under the skin to impact health and actually have physiological impacts. And Nancy Krieger provided us a framework to kind of guide us in understanding this in the public health community and titled it Biological Embodiment, in which she said that people literally embody biologically their lived experience in societal and ecological contexts, thereby creating population patterns of health and disease. And so my work is guided by Krieger's embodiment theory and seeks to elucidate these processes that she's speaking of. One of these being epigenetic mechanisms, as we mentioned earlier. Epigenetic mechanisms has garnered a lot of attention, and rightfully so, with thinking about epigenetic mechanisms and just giving a little bit of background, not going to go too deep into it. We're talking about outside of genetics. Epigenetic mechanisms impact the gene expression without changing the underlying DNA structure. Our body has over 200 cell types and tissues, all with the same DNA sequence. And it's the epigenetic mechanisms that are responsible for the cellular differentiation and programming. And so we want epigenetic mechanisms to occur so that we can differentiate our muscle cells from our cardiac cells, our liver cells from our nerve cells, so that we can have our organs and body systems working as they should. But what it is that is a concern is the aberrant DNA methylation pathways. And when these occur, that's what we're looking at as concerning to health. The main types of epigenetic mechanisms, histone modification and chromatin modification, which controls accessibility of DNA, small non-coding RNA or microRNA, which blocks transcription and gene expression. And the most commonly studied, and which I'm going to focus on today, DNA methylation. Oops. So DNA methylation, as I mentioned, controls gene expression without changing the underlying DNA sequence. This is basically when a methyl group is added to a specific site on the genome. That's the cytosine guanine dinucleotide. So when you add this methyl group here, it's either, this is where we see the methylation, DNA methylation sites. Many active genes have a cluster of these around their transcription start sites that features CPG islands, and most of them are unmethylated. In contrast, CPG sites that are between genes are usually methylated. In general, studies show that CPG methylation near the beginning of genes silences gene expression. However, it's important to keep context in mind, because this silencing of the gene, what does this mean for health outcomes? And CPG methylation is affected by what we know genetics, but also lifestyle choices and environmental exposures. And this is why it's become an attractive and plausible mechanism to look at as we link social environment to health disparities. And so the overarching goal of my work and what we're doing in my research group is understanding how the social environment influences and transmits health disparities across the life course, with a focus on epigenetic mechanisms. And so I'm going to now shift over and talk a little bit about some of the work that we're doing in my research lab. So important to the research that we're doing in understanding the origins of health disparities is the developmental origins of health and disease, or DOHAD, which acknowledges the importance of fetal life as a critical window for long-term health. Studies have shown that environmental factors can act during fetal development to program risk of adverse health in adulthood. We've seen this with outcomes of cardiovascular disease, obesity, metabolic syndrome, and some cancers. What's important to note here is, particularly to the developmental origins, is that during pregnancy, there are 3 generations present. There's the mother, the fetus, and the grandchildren through the germ cells. So there's the potential impact that environmental exposures can have across multiple generations. And so when thinking about the intergenerational transmission of disparities, we're wanting to really look at these exposures across time and see how these exposures might be passing down or transmitting disparities through possibly epigenetic mechanisms. And so as we mentioned earlier, I was funded through the National Institute of Minority Health and Health Disparities to really begin to dive into and look at these epigenetic mechanisms of prenatal environmental stressors on offspring cardiometabolic risk. So we entitled this project Prenatal Environment, Epigenetics, and Child Health Disparities, or PEACH. Specifically, this work is bringing together both the developmental origins of health and disease and the embodiment framework. And specifically, we are investigating how exposure to adverse social environmental characteristics and exposure to environmental toxins, both independently and jointly, impact offspring DNA methylation and offspring cardiometabolic risk. And the reason why we're interested in doing this both independently and jointly is because, as I mentioned earlier, due to segregation, racism, and these discriminatory policies, individuals who live in deprived and under-resourced neighborhoods generally are experiencing multiple co-occurring environmental stressors. So it's important to kind of think about how these multiple environmental stressors are having a cumulative impact on DNA methylation and offspring cardiometabolic risk. And so for this work, we're using data from the Newborn Epigenetic Study, or NEST, which is a racially diverse prospective birth cohort study with follow-up of offspring into early childhood and adolescence. This cohort is actually based out of Durham, where women were recruited from both Duke hospitals and Durham. At the time, it was Durham Regional, when they were between 6 and 42 weeks of gestation. At the time of enrollment, the women provided their addresses, which allowed us to geocode them and map environmental exposures. Currently, we have, well, right now, we have, I think just the police reported incidents data set. We have that mapped to the participant's address and kind of come up with some buffering and understanding the neighborhood presence of those, which I'll talk about. We're also capturing neighborhood deprivation using some information from the US Census and the American Community Survey. We're also going to develop measures of residential segregation. We also have information from the Environmental Protection Agency, or the EPA, on air quality, so small particulate matter, PM 2.5, and ozone. Of the 2,600 women who were recruited in the study, there was another project that was funded to capture or measure DNA methylation from the cord blood in the offspring. And so we have about 430 mother-offspring pairs with cord blood DNA methylation that we're using for this project. This, as I mentioned, is a racially diverse sample. We have 48% non-Hispanic Black individuals, 44% non-Hispanic white, 70% had at least a high school education, or the mothers had at least a high school education. We are following up with the kids. So we do know that the offspring, there's about a 32% prevalence of either overweight or obese children being classified as either overweight or obese, meaning having a BMI greater than the 85th percentile, of which 18% are classified as obese. And so during the study period, we received data from the Durham Police Department on police reported crime incidents. And so here I'm just showing some of the distributions of the violent crime during the study period in Durham. Durham has a weirdly shaped boundary, which is very interesting. And so as you can see from the map here of Durham, the violent crimes are concentrated in the city center, which is where the downtown Durham area is. And we expect that in urban areas, city centers which are more population dense, we expect to see a little bit more or a higher concentration of crime in these areas. What was interesting to us is how the magnitude in which these violent crimes were racially patterned. So the figure that you see to the right is the police reported violent crime incidents in the women's particular neighborhood. I think this is within 800 meters, so a half a mile around the participant's home. And we account for area because there are some issues of boundary effects. But what we're seeing here is if you look at the individual Black women and Hispanic women, we're looking at, I think this is 17 and 24 or 19 and 24 violent crimes during their pregnancy window. So I'm sorry, I want to say that this is during the gestation window that we're measuring these crime events, violent crime events. Whereas white women experienced on the order of about 3 to 4 violent crimes during their pregnancy. So if we're putting that into context, when we're saying that a pregnancy lasts about 39 to 40 weeks of gestation, we know some are shorter with preterm births and some actually go longer. But if we're thinking about a 39 to 40 week gestation window, a Black and Hispanic mom is hearing sirens, emergency response within a half a mile of their home every 1.5 to 2 weeks of their pregnancy. Whereas a white mom would experience it maybe 3 to 4 times. So for every 2 weeks to have this jarring experience of sirens, emergency response during one of the most critical and sensitive periods of your life, we imagine could be stressful, and that's one of the things we wanted to test. And so I kind of alluded to this in the last slide, but thinking of the stress mechanism of neighborhood violence and how this might be related to epigenetic mechanisms in DNA methylation, we know through the HPA axis and the release of cortisol, there is a stress response. And so we, using some sites that were identified in some previous work by Belinda Needham and Jennifer Smith, we looked at the association of neighborhood violence and DNA methylation at stress and inflammation specific genes. So in these specific genes, so BDNF, I'm showing here just the results from our, the significant hits. I will say, I know I'm showing some results with adjusted p-value. So this is in taking into consideration multiple testing that are above the 0.05. This sample size, because the NEST cohort, while they delivered at Duke and Durham Regional, they could live outside of Durham. And so we only had crime data for the Durham city limits. So we did have to restrict to just those living within Durham. And then, so that led us to a much smaller sample size, I think about 190. So we were a little more forgiving with the adjusted p-value. But what we see here is that we did find some significant hits for this higher level neighborhood stress variable on DNA methylation at 2 sites within the BDNF gene, which is a stress response gene. We did see a significant hit that's in the KLRG1 gene. This is an inflammation related gene, but we are going to interrogate the others that I'm showing here that are not highlighted that were moderately possibly associated with neighborhood violence. Because of our limited sample size, we weren't able to really do much with race specific effects, but what we are going to go back and do is just look at, for these sites, the average methylation among Black mothers and white mothers in our sample. Yeah. And so now moving a little bit further down the lifespan, our group also is evaluating the impact of neighborhood quality on epigenetic mechanisms, specifically epigenetic biomarkers of aging in adulthood. And so DNA methylation presents a unique opportunity to track changes in biological processes or aging and health risk and mortality. And due to array technology and machine learning algorithms, researchers have provided these epigenetic clocks or estimates of molecular aging using DNA methylation. So here I just show some snapshots of some of the ones that have come out, multiples come out. I feel like there's a new one every time I get ready to run an analysis. There's a new one that I could investigate. So we're looking at neighborhood quality in relation to epigenetic biomarkers of aging. In our research group, there are 4 that we are investigating right now, I'll say. The first is some of the first generation clocks and some of the earlier clocks. Hannum's clock, which was one of the earlier clocks to come out, which was trained in whole blood. And they identified 71 CPG sites that correlated strongly with chronological age. So these 71 CPG sites were used to provide an estimate of epigenetic age. Then Horvath's clock, which actually was tested and trained in multiple tissues, blood, skin, buccal cells, saliva. They identified 353 CPG sites that were correlated with chronological age. Lately, we've seen some of the next generation clocks that come out, Levine's pheno-age or phenotypic age, which has taken a different approach of not just looking at these CPG sites' relationship to chronological age, but also incorporating some important clinical phenotypes to get at the functioning or aging of biological systems and organs. So this includes glucose. It was trained on, I think, C-reactive protein, creatinine, red blood cells, as well as, I think there's 10 in total. And she identified 513 CPG sites that represented this phenotypic age estimator. And then one more recent is an epigenetic mortality risk score, which is a validated approach of estimating mortality risk using these 10 CPG sites that were associated with mortality. So when you receive these epigenetic age variables or these epigenetic age estimators, what do they mean? Well, we need to put them in the context of chronological age. And so to do so, we look at how the DNA methylation age differs from chronological age, or what is the residuals or the deviation from chronological age. And so we tend to look at this through positive versus negative age acceleration. So if your DNA methylation age is greater than your chronological age, we tend to say that that is a positive age acceleration or epigenetic age acceleration. So your epigenetic age is older. If DNA methylation age is less than your chronological age, it's considered negative age acceleration or epigenetic age deceleration. And as you can see, it's the deviations from this line of best fit that we're focusing on. And this just shows that for many of the age estimators, they've been associated with conditions across the life course in their various tissues, from Alzheimer's to cognitive performance, dementia, and even mortality. We've seen that these epigenetic age estimators are associated with aging-related health outcomes. And so, like I said, we're looking at how these are associated with neighborhood conditions. And so to do so, we're using the Detroit Neighborhood Health Study, or DNHS, which is a prospective representative longitudinal cohort study of predominantly Black adults living in Detroit, Michigan, from 2008 to 2012. And for this study, we were able to capture and measure neighborhood a little bit differently than what we have in the NEST study. We can actually look at both subjective and objective measures of neighborhood. Using, there's a quality measure that we created using 19 indicators. So we had, the DNHS study had trained individuals go out into the community and look at streets within block groups and assess the presence or absence of specific conditions such as graffiti, people on the street, alcohol marketing, smoking and tobacco marketing, and things of that nature. So we used these 19 indicators and tried to create measures of neighborhood quality based on how things cluster together using principal components. So we have principal components for neighborhood quality. We also have neighborhood poverty measures based on data from the 2008-2013 American Community Survey, which is the time that overlaps the Detroit Neighborhood Health Study. We also, because a person's own perception of their neighborhood is also very salient to their health, we have questionnaire-based data on neighborhood social cohesion. So using these 5 questions, we created scores for neighborhood social cohesion based on the close-knit of the neighborhood, willingness to help, you trust the individuals of these in your neighborhood, people get along, things of that nature. The sample size of this study is a little small because this was based on a pilot project at the time to assess DNA methylation in existing biospecimen that we have. And so we have 158 participants with neighborhood and DNA methylation data in whole blood. 87% of the population is non-Hispanic Black. 51% had at least a high school education. Flip that on its head. 49% of the population did not have a high school education. I'm sorry, 49% had more than a high school education. Whew. The mean age of this population is 54. Participants in this study were relatively long-term residents in their neighborhood, about 18 years on average. And the mean neighborhood poverty, or the proportion of individuals that live in the neighborhood that were under the federal poverty level, the mean for all of the neighborhoods that we looked at was about 38% poverty. For this study that I'm going to show here, this is a paper that was actually accepted 2 days ago, so that's exciting. We looked at Horvath's clock, Hannum's clock, and the DNA methylation phenotypic age. And here's just showing the correlations between the chronological age and the DNA methylation age. And as expected, because these were trained on chronological age, in our sample, they are pretty strongly correlated at a 0.8 or above. And as you can see, the red line indicates the exact matches, and it's the ones that are above the line as the positive or aging acceleration, and below the line being the negative age acceleration or deceleration. And so I'm going to present here just some of the findings from the principal component analysis. So there was one principal component that was associated with epigenetic aging across all 3 of the aging estimators. And that was one characterized by the presence of abandoned cars on the streets, as well as people on the streets. So kind of getting at some measure of disorder, neighborhood physical disorder. And so what I have here is a figure where the effect estimates are on the y-axis and the different epigenetic age estimators are along the x-axis. So the estimates in the boxes, these are for the total sample. The triangles are for women, circles are for men. And what we see here is that in the total sample, there is some indication that poor neighborhood quality or more disorder is associated with epigenetic age acceleration. And it seems to be, especially with the pheno age, some suggestion that this might be driven by the association in women. So there's, we can see here that the effect estimate, or the triangle for the pheno age, is in the order of about 2 for women, where for men it's relatively null or 0. Interestingly, when we look at now neighborhood social cohesion, we see something totally different. For men, neighborhood social cohesion, so living in a neighborhood with more cohesion, is associated with an epigenetic aging deceleration. So something about the positive aspect of the neighborhood is possibly protective for the men in our study. And so we were interested and thought this was really cool. So we were like, well, how is there something that potentially can happen when for individuals that live in disadvantaged or poor quality neighborhoods, does social cohesion offer some type of benefit? And so what we found was, yes. So this figure here is now showing the results where the triangles and circles represent the stratification on neighborhood social cohesion. And this is the models for neighborhood quality. So that principal component with abandoned cars and people on the street. Here, again, the squares is the same results that I showed previously on the first figure, where we see neighborhood quality being associated with epigenetic age acceleration. So higher neighborhood physical disorder associated with epigenetic age acceleration. But what's interesting is when you stratify this on social cohesion, the association only remains for those who live in neighborhoods with low social cohesion. In other words, living in a neighborhood with high social cohesion, or the triangles, we see that association is attenuated to being no longer significant. And so there seems to be some evidence that there might be, and I am saying this very suggestively because we only had 158 people in the study, and I know the confidence intervals overlap, so we can't really dig too deep into it. But I will say that the association remained significant for those in low social cohesion neighborhoods, whereas for those in high social cohesion neighborhoods, it did go to null. And in another study that was published last year using these same data and the principal component analysis for neighborhood quality, we also saw similar findings with epigenetic mortality risk score. So here I'm showing principal component 7, which was characterized by abandoned cars, people on the street, non-art graffiti, and the street being in poor condition. Again, some indication of disorder, neighborhood disorder. We saw, again, some sex-specific effects where the association with increased epigenetic mortality risk score was only shown in women, not so much for men. And also that there possibly is some benefit of living in a neighborhood with some positive attributes. So here we are stratifying on green space, so the presence of trees and gardens versus having low trees and no gardens in the neighborhood. And what we see is that the association is attenuated. So the green box, it's attenuated to null for those who live in neighborhoods with green space, so high presence of trees and gardens. Whereas those that have low trees and no garden, we see that the positive relationship between poor neighborhood quality and epigenetic mortality risk score remains. So again, giving us some evidence that there might be something to say about positive attributes of the neighborhood. But again, I will say that study was limited in sample size as well. So we do plan to replicate this in a larger cohort, possibly AdHealth, or the National Longitudinal Study of Adolescent to Adult Health. But to bring together the projects that I described here, neighborhood social environments appear to have some effect on DNA methylation in early life periods and adulthood. and that there's some indication that positive community features like social cohesion and green space may provide some protection. And I just want to park right here for a second and say that this is, to me, a very important finding, because as researchers, we tend to go into this work thinking of neighborhoods in a deprivation or disadvantage or a lacking lens. But what we see is that even in these neighborhoods, there exists some fabrics that might be protective against or buffer some adverse health outcomes. And so when we're thinking about interventions and how we can actually reduce health disparities or create health equity, really looking at neighborhoods not from this disadvantaged lens, but also looking for the positive attributes that might provide some protection. And so where do we go from here? As I mentioned, we're planning to replicate these findings in larger cohorts, because I think that would be important to substantiate these results. Also, all of these studies that I showed here, the work that I'm showing here stops at epigenetic mechanisms or the DNA methylation findings. It's important to relate this to health outcomes, because with the health epigenetics, what does this mean? So we plan to, in both studies, relate this to health outcomes, investigate this in relation to health disparities. One day, hopefully, particularly with the AdHealth study, even relate this to gene expression through one of our recent grants that we have. And also how this relates to health disparities in childhood and adulthood. And so this brings us to back to the overarching goal of the research group, is really understanding life course social epigenomics and the independent and joint impacts of prenatal environmental stressors on DNA methylation and offspring cardiometabolic health disparities. As I mentioned, I only showed the crime results, but we're also interested in looking at the intersection of the neighborhood crime deprivation with the air quality measures that we have as well, and how that relates to offspring DNA methylation and cardiometabolic health. With the AdHealth study, we are able to look at early life social stressors, as well as across the life course, and how that relates to adult DNA methylation and health outcomes and health disparities, particularly with cardiometabolic health and even some mental health indicators. And where we hope to go with this, particularly, with the NEST study, is we're following up the kids now into adolescence and capturing some biospecimens, so that we hope to be able to measure DNA methylation at a second time point to begin to look at, from this developmental origins of health and disease framework, how do epigenetic marks change as individuals' environmental stressors or their exposures, environmental exposures change. So going from the fetal environment exposures to the child actually experiencing their own environmental exposures, how do the DNA methylation patterns change? And so with that said, I'd like to acknowledge my amazing research collaborators and team, who without them, this work would be impossible. I also want to shout out the fact that this team is very interdisciplinary, from researchers in sociology to individuals working at the EPA and environmental sciences, from undergrads that were working in my research group to faculty members that are contributing to the research group. The amazing study participants, without them, this work would not be possible. The funding sources that have contributed to this work and allowed us to really dive into these questions that related to social determinants of health and disparities. And with that, I'll stop and open it up for questions. Thank you.
That was fantastic. Thank you, Dr. Martin. If anyone has any questions, you can raise your hand. I'll call on you. You can unmute yourself. You can also put it in the chat and I'll read it out for you. So there was a question about what the what the measure people in the street is. Does it mean people walking around like pedestrians or loitering?
So it's more loitering. They were asking about, are people loitering, standing around, kind of this disorder. So the evaluators were trained to kind of look at things that were abnormal to, I wouldn't say abnormal because it's normal to their, but just disorder. I'll say that.
Bree, you got a question?
Yeah, actually, I do. So I wanted to know if I know you mentioned that you're looking at these marks in like really early stage development, like in young individuals, but like, do you think that there's a certain amount of disorder that you can experience within your genome, right? Like epigenetic modifications that once you're older, that would eventually like maybe disappear from that? Like where would you, I guess you want to do this more longitudinally, like where would you remeasure, like at what point in someone's life?
Great question, great question. So we've been discussing this in our research group because we have the NEST cohort where we have their DNA methylation marks in cord blood, so kind of this offspring marker of DNA methylation. We also have these early neonatal markers through buccal cell and saliva, but we're also following them and we'll have them in adolescence. And so one of the things that's interesting when you think about the developmental, in this early stage developmental period of and stress of delivery and what's going on with the baby at delivery with the DNA methylation. So epigenetic marks, we tend to, and what I've seen in literature is it takes a while for them to change, but the developmental period, so from that fetal environment, because it's your mother's and your in utero environment, you're not actually experiencing it yourself. We do suspect that once a child is old enough to begin to experience their own environment, because again, and this is now putting on my epidemiology hat, it is the question of the shared environment versus your own environment, because in most cases, young kids experience the same exact environment as their mothers or their parents or whoever they live with. So we want to give enough time for them to allow us to see some differences or discordance in environments. So I would say that the NEST project that we're planning to do, we will capture and look at the differences, see if there's changes in adolescence from what we saw in the cord blood. So that's where we would try to get at the longitudinal piece. Ad Health, which I didn't plug Ad Health, but I want to plug Ad Health now. It is probably, in my experience, one of the most rich data sets. It has, these individuals were recruited when they were in grades 7 through 12, and they have been followed. Now we are actually beginning to start developing wave 6 when they will be in their 40s. And this is 15,000 individuals. So it started as a nationally representative sample. And there are biological markers, biospecimen. There's some blood spots that were captured in young adulthood. So we might be able to do some longitudinal measures. We have a social epigenomics project right now where at wave 5, when they were in their 30s, we are measuring DNA methylation. We hope to be able to do that again in wave 6 when they're in their 40s. I don't know how much I suspect to see changes there. For the most part, DNA methylation tends to be a little stable, but it'll be interesting. I think one of the things I like about this area of research is it's still evolving. So it's one of the times that I can really say, I don't know, and really mean it. We don't know as a community.
Thank you.
You're welcome, good question.
Kaylin, you got a question? Go for it.
Yeah, hi. That was a great talk. Thank you. I'm wondering if you've come across or heard at all about, I think it's called STAND, in Detroit, and it's I believe it's a study where they're setting up green spaces, sort of randomly matched neighborhoods.
Oh, wow.
And because I think that finding of the green space finding is so exciting, and it's so interesting. And I think certainly amidst the pandemic, all of us have had our different experiences, both positive and negative, with green space or lack thereof. So I wonder, I think it sounds like that might be a really interesting example where you might be able to start to really look at the idea of green space as sort of a buffer, as you were talking about. I think that would be really cool.
Absolutely, thank you. And that was called, what did you say it was called?
I believe the acronym is STAND. I think it's looking at cancer health disparities.
Okay.
And I was sort of talking to some people about that, doing some stuff there, but I guess I don't know if you might have better connections there or not.
I will ask, I think, yeah, that would be, I mean, that's, and even thinking about what you just said of like randomly putting them up and being able to look at neighborhoods that were assigned and green space was developed versus those that were not developed, green space was not developed in, and seeing how that impacts. Yeah, that would be tremendously interesting. There it is right there in the chat. Thank you.
Yeah, and it seems like it's Detroit is almost the perfect place for it too, just where it's at, I think, socially, culturally right now.
Exactly. Thank you.
Bree, you had another question?
Yeah, I was just wondering, I mean, I know you found the green space situation as a potential buffer. Have you guys thought about any other potential buffers that could mitigate some of this epigenetic?
Yeah. So a little backstory. I actually got involved in epigenetics because I was thinking about diet with methyl groups and methyl donors. Could diet be an intervention, or, healthy diets be a way that we buffer these effects? But then, my social epidemiology diet was, it has its own issues, access to diet and healthy foods and those sorts. And so, we have thought about lifestyle factors. I really think the perceptions of your neighborhood. So, not just what we see or what the census reports as poverty, but when I've been talking to people who live in neighborhoods where there's a high presence of crime, there's also individuals who say, I love my neighborhood because there are grandmothers that live down the street that are all looking out for the kids. And so that fabric of cohesion and collective efficacy, I think is really interesting, and one that I want to kind of explore a little bit more with a larger sample size, to see if it sticks.
Thank you.
Thank you.
Other questions? I want to give space. Liz, go for it.
Hi, Dr. Martin, this was wonderful. Thank you so much.
Thank you.
I was really excited to hear about the measures that you're developing. You indicated that you're developing some measures of residential segregation when you were talking about the project in Durham. And I'd love to hear more about that, because I know that often with previously collected data or state collected data, the questions asked maybe slant towards structural racism, and how can we develop better tools?
Oh, yes, thank you for that. So traditionally, what we have found is from the okay, let me back up. So we have the census data, and so traditionally what people do is when they want to develop a measure of residential segregation, they just look at the composition of the neighborhood. Is it 75% or more Black, or 75% or more Hispanic? But that only tells us so much, because what does that mean in the context when you're in a neighborhood, when you're in a community like Detroit or somewhere where, it's a pretty homogeneous area? So some of the more recent measures get at there's the dissimilarity index, isolation index, in which captures how many, what proportion of Black individuals or white individuals would have to move out of that neighborhood and into another neighborhood in order to integrate or make that a less segregated neighborhood. So that's something that I think of is of interest in that we're exploring. We also are looking at, and I will say, the students right now are doing tremendous work of just like diving in of how can we measure it based on administrative data. A lot of times with the population data sets that we have, we're limited in what we can assess for these area level measures. And so with the residential segregation, they're also thinking about, there's this, index concentration of extremes. So an ICE measure that is used that we could also look at dissimilarity or kind of get at segregation. One of the things that, and I want to take this time to talk about the crime piece too, because I think that's also really important when we're thinking about segregation. One of the things that we, when I went into this study and I wrote the proposal, I was like, I have data from the Durham Police Department on crime. I'm going to look at this. And as we begin to look at this and because I have sociologists and folks on my team, we started to realize crime is a product of two things. It's the actual incidents and it's the presence of policing. And based on the neighborhood makeup, you are actually going to see higher incidents because there's just police there. I didn't present the data, but when you see the disparity or the differences in the distribution of drug-related crimes, it's astounding. Again, it's concentrated in the city center, but we know that that is primarily due to age-old policies like the war on crime and all of that stuff, where police are sitting and, surveillancing certain communities, but drugs happen, or people are doing drugs at similar rates, comparable rates across, so why do we see these differences? And so now we're even trying to develop measures of policing that get away from really focusing in or emphasizing or utilizing the police reported crime measures, because we know that that is a product of where police are. So we've gone back to the Durham Police Department and we've gotten arrest data, we have calls for services data, so that we can start to merge all of this together and see, based on the makeup of this neighborhood, really what is going on in terms of policing versus crime. So yeah, there's so many things related to segregation, so many ways that we are measuring, but I do think we have to go beyond and kind of think critically about what is provided to us in these administrative data sets and how can we tweak these to really get at the constructs that are important to structural racism. So yeah, thank you for that question, Liz.
Thank you, Dr. Martin. I just think it speaks so nicely or dovetails so nicely to the social cohesion index that you had in Detroit, and it just, opposite ends there, but really powerful. Thank you again.
Thank you.